Welcome. I hereby open this academic session in which Antonio Criscuolo will defend the academic thesis, Ordo ab Chaos, Timing, Cognition and Waves. May I invite you to present your summary of your study and the conclusion of your thesis.
Highly esteemed Pro-Rector, members of the opposition, supervisors, family and friends, thank you for joining me today, either in person or online, for the public defense of my dissertation. As the title and the beautiful graphics suggest, today I'm going to be talking about waves, and I'm going to be discussing how waves can generate an emergent sense of time. In my research, I'm particularly interested in understanding how we process the when, the precise timing of sensory events in the environment, and how we generate predictions about the when of future events. Drawing a parallel with physics, I argue that we generate a sense of time using waves. Waves allow to discretize the continuous flow of time into finite units, oscillations and oscillatory cycles. What is also interesting is that we can also use waves to characterize physiological activity in the body and in the brain. And so, as we shall see, these body-brain physiological oscillations allow for perception and action. In fact, I'll try to convince you that this emergent sense of time allows multisensory input to converge into a temporally coherent perceptual representation. My dissertation is a journey from pre-Socratic philosophers to Newtonian physics. I discuss concepts of time from Aristotle to Einstein, and I do so bringing Pino Daniele to the Amalfi Coast, meaning I provide very concrete examples with things that I like on which timing operation we need to understand the world. Why do I care so much about time? Because me, as many others before, think that our lives are governed by the passage of time. Every heartbeat, every breath we take, every decision, every action happen in time, with very precise timing at multiple temporal scales. Our music and speech and other animal behaviors enrich the environment with gradients of temporal regularities. In fact, in our environment, we can characterize rhythms. For example, we can quantify the number of steps while walking and running. We can quantify the number of syllables while speaking and singing. Similarly, we can quantify the refresh rate of the screen that you have in front of you. And if we extend this axis on the left side, we can even describe cosmological regularities, meaning the spinning of pulsar stars, rotation of galaxies. Now, what I've been doing is actually describing environmental rhythms in terms of frequencies, so occurrences per second. We can complement such description by also plotting the amplitude of such events. And now what we're looking at is a Fourier spectrum. We have now learned that we can use such analysis, Fourier analysis, to quantify regularities in the environment. What is also interesting is that we can use a similar approach and similar graphs to also characterize physiological activity in the body. In fact, we can characterize individual heart and breathing rate, but we can also quantify regularities or rhythms in brain's electrical activity. In fact, oscillations in the brain can be clustered in frequency bands. So there are faster oscillations in the gamma band, and there are slower oscillations in the delta band. Now, little summary, we can describe periodicities in body, brain, and the environment with such type of analysis, Fourier. We're going to see these graphs later on. And the next step is actually saying that what we are interested in doing, it's also looking and quantify the correspondence between these environmental rhythms and body-brain physiology. In such a way, we think we can understand something about timing operations in the brain. What we need to do to characterize mutual relationship between oscillators is parametrizing the waves. So we can take the example of a pendulum clock, and we take the pendulum trace oscillations on the time plane. Now, these oscillations have a few characteristics. They have an amplitude, alpha. They have a period, which gives us the frequency, how many times it repeats per second. And then there is the phase. Now, the phase is of fundamental importance. We can think of the phase as this green dot that serves along the wave. We can think of the phase as giving us an information about the exact state of the oscillations over time. And because these oscillations will repeat periodically over time, we can either describe the phase as this continuous line, or we can wrap the phase around a circle. Remember the circle because it's going to be useful later. Now, why do we need the phase? We need the phase because with such an information, now we can really quantify dynamics of oscillations, but we can also characterize mutual relationship between oscillations. So these oscillations can be in phase, anti-phased, aligned, or they can be phase shifted over time. And we can use it to characterize how oscillations may be tuning towards towards a convergent mechanism of alignment. Now, this is important because we actually think this is how the brain processes environmental rhythms. Namely, for example, when we are processing speech and music, we tend to align neural oscillations to regularities in music and speech. And so these metrics will be fundamental for our understanding of timing. Okay, now we have a little bit of trigonometry, we know Fourier analysis, we know oscillations, and we can start asking a few research questions. So, in my research, I try to address a few fundamental questions. How do we process the timing of sensory events? And how do we use timing to coordinate perception and action? To address this question, I look at the neurophysiological mechanisms to encode, track, and predict timing. This means that I look at brain electrical activity as recorded with EEG, electroencephalogram, and I search for the building blocks for our capacities to detect, produce, and synchronize with environmental rhythms. This is the working framework, and then in the chapters, I ask specific research questions. In chapter 2, the first research question is, can we identify a neural code for timing in healthy humans? In the next chapter, I ask whether there are similarities in the neurophysiology of basic timing processing between humans and macaque monkeys. Next, I ask whether lesions in the basal ganglia and the cerebellum, fundamental subcortical structures for rhythm and timing, impact our capacities to detect, produce, and synchronize with rhythms. And finally, in the last chapter, I ask whether such interest in rhythm and timing can be extended beyond the brain. So is there an influence from the body on our emergent sense of time and in general cognition? Overall, in my research, I take a basic comparative and translational approach on timing, meaning we take an evolutionary perspective on these basic timing capacities and potentially their role throughout evolution, but we also address questions on what happens when there is altered timing in the clinical population. And so, with these questions in mind, what do we do in the lab? We record EEG, electrical brain activity, while people listen to very exciting and grooving auditory sequences. These are metronome presented at a specific rhythm 1.5 hertz. That's what they're doing. And with such an easy setup, then we ask the question of whether the temporal regularity in the acoustic sequence is reflected in neural oscillatory activity, these waves over here. Now, we have seen it before. The first way to address this question will be to run Fourier analysis. And here we quantify the amplitude, the strength of the peak at the stimulation frequency, this 1.5 hertz, which corresponds to the auditory sequence that you were listening. And next, we want to do something else. We want to quantify whether these neural oscillations tend to align towards expected onsets whenever you are hearing a sound. How do we do that? We look at the phase and we quantify coherence via circular statistics. Little summary. What we see is that delta waves encode timing with such type of analysis. We quantify the phase alignment of neural oscillation in the delta band to external sounds. But then we can also quantify intra and inter-individual variabilities in these mechanisms of alignment. And this becomes of fundamental importance, especially when working with patients. So, what did we find in our research? In chapter 2, I describe a delta beta neural code for processing and predicting timing. What I show is that slower delta oscillations, but also faster waves, beta oscillations, are complementary in creating this emergent sense of time or in processing the timing of the auditory sequences. In chapter 3, I show that there are striking similarities in the neurophysiological signature of basic timing processing. So here we had macaque monkeys and humans, and we show that there are very similar neural mechanisms in place. And in doing so, we potentially also draw a link between this basic timing function and development of higher order cognitive functions in humans. In chapter 4, we show that lesions in the basal ganglia and cerebellum impact these fundamental capacities to detect, produce, and synchronize with rhythms. In fact, these patients not only couldn't process temporal regularities in the auditory sequence similarly to healthy controls, but they also didn't manage to synchronize their behavior to the external rhythms. And finally, in chapter 5, we extend this interest in rhythm and timing with a novel framework for a body-brain dynamic system. So the position here is that maybe the brain does not work in isolation, but body physiology exerts a fundamental influence on brain activity and shapes cognition, from health to pathology. What we are arguing there, first of all, the starting point is saying that we have long neglected modulatory influence from bodily physiological activity. What we should rather do is embracing the complexity of these bidirectional interactions. And how would we do that? We quantify dynamics of these oscillations over time. So we can characterize states of synchronization, desynchronization, but more importantly, what we should do is actually characterize dynamic state transitions between these coupled, decoupled states. Why do we think that is important? Because we believe that these body-brain dynamic state transitions serve the scope to optimize perception, sensory processing, attention, but also motor coordination. And it is in our daily experience that we use mechanisms of, for example, top-down control of breathing for improving motor coordination, when speaking, for example, but also we try to coordinate breathing in order to improve auditory processing when we are in a noisy environment and we try to listen to something. All right, this brings me to the summary and conclusion. I started by discussing that time is a fundamental dimension of our lives. We use this sense of time to act and adapt in a dynamically changing environment. I showed you that we can think of timing processing by looking at neural oscillations. And in particular, we identify this delta-beta neural code for processing timing. I showed you that there are similarities in the basic neurophysiology of timing processing. So this was a comparative study with macaque monkeys. And in doing so, we also potentially lend support to the role of timing throughout evolution for adapting to the environments. Then we showed that the basal ganglia and the cerebellum play a fundamental role not only in timing, but also in the capacity to produce and synchronize with external rhythms. And the truth is, in these studies, there is much more. So I hope the opponents will ask questions on more results and more data, because I didn't have the time to show here. And then we have this novel framework for a body-brain system, with the idea that if we really want to understand perception and action, then we should look more holistically at how also the body influences cognition, from health to pathology. And because I have the last minute, a personal note. In 4 years, there are many things happening, but overall, I mean, PhD is a fantastic journey. And it was also because I'm surrounded by super exciting researchers. And I had two very motivating supervisors, always encouraging me in every steps and motivating the next steps. In fact, there is a lot going on and planned. It's also an opportunity for traveling. I probably did too much. Sonia didn't like it, but you know, it's fun. And many people travel to my own hometown for this conference that we started organizing. And look how happy they are in Amalfi, right? And then, you know, other little things, you know, we grew up, so we have to make also other steps. I want to always thank family and friends for surrounding me. And I want to thank Elena. with this, I thank you for your attention. I'm really looking forward to discussions. Thank you.
Yeah, dear candidate, thank you very much for your presentation and personal additions. We now open the opposition with the chair of the reading committee, which is Professor Dr. Bernadette Jansma, who is chair for cognitive neuroscience of language here at Maastricht University.
Yeah, thank you, Pro-Rector. Dear candidate, first of all, let me tell you that I enjoyed reading your book very much, and I congratulate you and also your supervisors to this great work. I think what you did, you mentioned this already in your thank you, so I can be short, but actually what you start up nicely is fundamental research, then you do cross-species comparison, and then you even go into translational cognitive neuroscience working with patients. It's very impressive and I think very relevant on a certain concept, namely how do we perceive the world and what is time as a how relevant is time for that? And that actually brings me to the question. I also should mention that you published well, that you went to many, many conferences, that you even are editor in book chapters already, and that you are organizers of brainwave conferences at very nice places. And I think keep on doing it this way. But the question now is, in your beautiful introduction, so I really read that with pleasure, you also here you talked about the sense of time, but actually I think your work is on a time of senses. You get the difference, sort of?
Can you elaborate?
Yeah, so there's the perception of time or the timing of perception. Yeah. And I wonder, so you did this, you came up in the last chapter with this integrative model where you actually use or make use of regularities in body, brain, and behavior and combine that. And your assumption, you also mentioned it in the introduction, is that time is regular. But I severely doubt that. Being a psychologist, time is not at all regular. So if I go the stairs up to third level, sometimes it takes me for ages to get there. And sometimes I'm there in a sec. So I have a subjective divergence in how I experience time it takes me to do something. Might be related to my inner state, my bodily... Same as in perception. I think in visual perception, people are very outspoken about this and say that they did actually work on it. On a basic paradigm would be you see a sequence of pictures and you estimate the duration of the display of these pictures, and people vary in how they estimate the duration, and they vary based on the complexity of the stimulus content. So time is not a constant, at least not the perception of time. And hence, I wonder how you could incorporate this variation in the perception of time into your model.
Yeah. Highly esteemed opponent, thank for the beautiful question, which encourages a very long answer. If I could, I would. Yeah, but I'll avoid it. I mean, as you noticed in the thesis, I start from conceptualization of time because indeed I start then discussing that time is not fixed. But what if also this sense of time is not fixed and it varies across individuals? So there, yes, there are a couple of more philosophical type of discussions on how we can actually even discretize time. How do we think of time? But then I always talk of a sense of time because we can think time as a tool for understanding the world rather than something fundamental. And we know also from the laws of physics that time is not fundamental, it's emergent. And I tried to say emergent a couple of times also during this presentation. And we know that from more classical, if you want, physics, that we use time for space and motion, but we also know from quantum mechanics that actually time is not fundamental but emerges from the super... Anyways, okay, I'll go to the brain part, I guess. Okay, in the brain. So indeed, I try to speak of a sense of time that is influenced by this body-brain type of coupling. And in that sense, then anyone creates a sense of time that varies even during the day, but surely varies across individuals. how can we characterize, understand these inter-individual variabilities? Are they actually important for understanding cognition? Is it useful for understanding pathology? Hopefully so, but I don't think that there is too much exploration on the variability or these timing distortions, no, for actually understanding the disorders of any cognitive functions.
Yeah, I shortly interrupt because we really also are urged to be. That brings me basically to a very simple question, I hope, is if you elevate or modulate the complexity of your stimuli, would you then assume that in your timing, tracking, cross-frequency coupling of delta beta, you would assume shifts in the frequency or shifts in the phase coupling?
Definitely. So indeed, most of the times we use these simple isochronous sequences because we don't want to have any doubt. The regularity is there. We want that regularity to be reflected to some extent in neural oscillatory activity. And any variability there, it's either informative of something else that happens, so this theta-beta type of phenomenon. We see subjective preferences for superimposing accentuations, chapter 2 and 3. But then in chapter 4, I try to use a different method, that it's actually about the tuning of oscillations to some perceived regularity, if you want. And that it's nice because indeed you don't see these oscillations always tuning to the temporal regularity, but you see that they fluctuate over time. And the variability in those fluctuations can actually even distinguish the patient population from the healthy controls. And it's not only the variability, we use all these metrics, probably we did it too much, but it's shown in the entropy, in the acceleration, in the stability, and so on and so forth. So yes.
Okay, thanks. Time's up. I give the word back to the rector. Thanks.
Yes, thank you. The opposition will now be continued by Dr. Vincent van der Ven, who's Associate Professor, also at Maastricht University with expertise in time processing and EEG methods.
Thank you very much. Dear candidate, I very much enjoyed reading your thesis. It's on a wonderful topic, which I think a lot of more people should focus on because it has implications for a lot of different behaviors and cognitions. I wanted to comment that I thought your writing style was reminiscent of Carlo Rovelli. I'm sure you know your Italian countryman, who also wrote a book about time. Your main premise in your thesis is focused on oscillations, and my questions would be particularly about that. So, in your conclusion statements, or in your last chapter, you write that essentially, or that you showed that we obtain our sense of time from oscillations. And I assume that you state that oscillations work like some sort of clock. And I have some questions about that, because on the one hand, there are those that would say there aren't really any oscillations happening in the brain. It just looks like something that oscillates. But even if you would consider that there are things happening regularly, the oscillations that we observe in these type of studies, like in yours, are often quite variable. They vary in phase, they vary in amplitude, and they can also wax and wane or even disappear. And there's of course the problem of so many different oscillations at different frequencies that one wonders if there is a clock or many clocks, how does this mess actually lead to a sense of time? Yeah. So is it really a proper model to account for a sense of time? And maybe these oscillations are perhaps representing something else that, although temporally coded, doesn't code time.
Highly esteemed opponent, thanks for the question. Nice one. Indeed. Also here I would maybe exaggerate, but so let me start by saying, so I think the beauty of chapter 2 and 3, it's actually going a little bit, against the typical finding where, these oscillations do encode regularities, they tend to align perfectly, phase as an effect, and so on and so forth, in such a way that we show that there is a lot of variability, which usually is not described. There is variability, obviously, across channels for, volume conduction and so on, but there is lots of variabilities over trials. And when you don't take the typical averaging type of approach to these trials and so on, you cannot indeed identify such, beautifully coded alignment mechanisms. So indeed, the question is there really an internal clock that tends to align to these regularities, or these oscillations are epiphenomenal? So yes, on average, they look beautiful, but on a trial level, there isn't too much happening. So probably it's a sequela of evoked responses, probably it's resonance, because there are some activity that seems to indeed resonate or somewhat oscillate at that specific frequency. I think that is the open question, and I think these chapters only motivate more questions on investigating that a little bit better. What I try to do with chapter 4 is indeed saying, okay, if we want to describe real oscillations, endogenous oscillators that pre-exist and outlast the simulation frequency, then we need to use different methods. We show that these oscillations are before the sequence and they are after the listening period, but then there is some tuning in and out of the auditory sequence. If such thing happens, then okay, maybe we do have some form of oscillations, but if that such thing doesn't happen, meaning we have somewhat of a flat line, and then we start seeing some evoked responses that look like oscillations, then those are not oscillations. Do that method really convey now a novel take on oscillations? I don't think so yet, because obviously we use frequency filtering or wavelet decomposition and so on, which by themselves impose oscillations. So, okay, chapter 5 comes into the game, right, and says, okay, maybe all that variability that now we are observing at the single trial level and all this inter-individual variability in this tuning mechanism to very simple isochronous sequences may be due to also fluctuation in arousal states from the body. So can we characterize variability in arousal states from the body based on breathing, based on cardiac patterns? But also there, it's not a regularity. What I try always to say is that these are oscillators that also show dynamics, right? So maybe the heart, if it's a real clock in the body, should also show this tuning in and out of some regularities, or at least should be reactive to the environment if it's influencing for real the brain processing of these regularities. So maybe the short answer is there is so much to do and we don't know the answer, even how to address oscillations, right? There are so many methods, and the truth is there is variability, not often described.
I fully agree. However, is that a good basis to actually consider that this phenomenon would actually arise to a sense of time? You could also argue that these oscillations serve a very different purpose, such as, for example, predicting a next state that doesn't, that doesn't have to convey, let's say, temporal duration. It could just mean that one item predicts something else to happen, and that would be about it. So you don't reach a sense of time out of this, as you phrased in your thesis.
And in fact, there is a beautiful paper that from time to time I read and reread. It's from Asaf Breska and Leon Duell, 2017 in Life. And they're like, okay, oscillations, yes, they potentially show what people call, temporal predictions, but what if it's predictability? In the sense that, indeed, over averages and so on, they show a mechanism of alignment, but it's a memory-based type of alignment rather than real predictions. I don't think that, it's not that I don't think, but it's very hard to show, real predictions from predictability. But yeah, there are a few papers from those authors there that actually, have nice paradigms for doing it. Yeah.
Okay.
I give it back.
Yeah.
Thank you very much.
Thank you.
Yeah, thank you for the questions. We continue the opposition with one of the members of the opposition who is joining us online today. That is Dr. Anne Keitel from the University of Dundee, with her expertise being in auditory and speech processing. The floor is yours.
Hi, Antonio, and thank you for this wonderful presentation. I also thoroughly enjoyed reading your thesis, and I have loads of questions, honestly, so many questions. But of course, we don't have a lot of time, and I had to narrow it down a bit. And I'm sure we'll maybe have time at the WAVES conference in September to talk about your work. So my first question is about your dynamic body-brain system theory, where you say that a decoupled state between cognition, behavior, heart, breathing, and brain activity, that's the default. And of course, if there's no behavior, then there's nothing to couple to. So there can't be any coupling with behavior, but the heart, breathing, and brain activity could well be coupled during rest. And I think that's what some emerging research has been showing. So I wonder where your starting point came from. What do you make of this new research that, for example, breathing is coupled with the brain?
Highly esteemed opponent, thank you for the question. Nice to see you. So yeah, I think what we wanted to do in chapter 5 was indeed taking a slightly different perspective, but it was more like providing a framework within which we can develop new theories and potentially, you know, methodological approaches. Indeed, the core point is that we not necessarily need to quantify these correlations over time between, or these coupling phenomena between different bodily rhythms and brain activity, in terms of saying, okay, if your heart rate is 1.5 hertz, then your center delta band frequency is 3 hertz, and so on and so forth. So creating such a frequency architecture that it's just based on harmonics, and it's somewhat of a static architecture that I don't think informs too much the variability of any type of daily behaviors. Then the idea is indeed dynamic state transition. So we want to see what happens over time, and if there are adaptive mechanisms in place to coordinate action and to optimize perception. With the idea that, yes, in clinical population, we may be able to characterize some altered patterns of state transition, or maybe we don't manage to characterize such state transition, and so there isn't optimization of perception, there is actually, you know, impairment of sensory processing and so on. Where did we start from? I don't know, it was just like a brainstorming and so on. There is obviously, you know, plenty of literature by now, and it's growing interest. That's why we're also organizing these conferences. But most of the times, indeed, it's hard to find this continuous matrix of coupling, and it's surely very difficult to find metrics of variability. So that is why we thought, okay, now we start generating new questions, potentially with this idea that you characterize states, always with the idea that you have these high versus low excitability states, at least in neural oscillations, which can be modulated by arousal states of the body. Was there a second question? I don't remember. Maybe I lost it.
Yeah, no, but I have one now, also based on what you just said with the excitability of neural oscillations. So in your first study, you do have a lot of analyses, and I really like the analysis that you looked at the delta phase and how that varies within trials and across trials. I think that was like long overdue that someone did that, so that I really like that. But what you do show is that the delta phase during listening to those isochronous tone sequences is variable across trials, which conflicts with the idea that we have an optimal phase. So this idea of excitability and less excitability or inhibition is that, you know, we have this optimal phase to bring upcoming stimuli in an optimal window for our processing. But your finding directly conflicts with that. So how can you reconcile this with the idea of the optimal phase?
So I think, you know, probably I mentioned it before, that it was a little bit of a challenge to whatever we see in a little percentage of nicely published papers where they see this optimal phase effect, in humans at least, with non-invasive recordings. But methodological differences. So here, for example, what I'm trying to do is focusing on the pre-stimulus time windows. So what I'm doing is looking into 60 milliseconds before the stimulus onset. I'm not looking as usual at the zero, so at the tone onset. With the idea that in those 60 milliseconds, if there is any mechanism of predictive phase alignment, then I wanted to quantify somewhat of a build-up or a tendency towards alignment. So somewhat of a, you know, a motion towards optimal phase, which does not happen. I explore a frontocentral cluster with 16 channels, trying to see whether, you know, there was maybe one channel that is doing, another channel is doing in a different way. I mean, it would have even been nice to see that, you know, different channels achieve different sort of, you know, phase alignment at the onset, and maybe it's explained by volume conduction and so on, but it doesn't happen, right? So, okay, in some trials, then we do quantify this mean vector length as a metric of coherence. So in some trials, the alignment happens. In some other trials, which is actually a majority of the trial, does not happen. Is that fluctuation due to fluctuations of attention, engagement in the listening, and so on and so forth? I don't know, but we currently have a study where we are trying to see in a continuous listening, so it's more than 10 minutes of listening to these, groovy isochronous sequences. And there, okay, we are expecting this tuning in and out, or like flowing in and out of attention. And we want to see if attention, using a proxy of alpha power over time, can inform not only delta band power, but actually delta band phase coherence, with an intertrial phase coherence with moving windows over time. Such cross-frequency coupling mechanism may potentially, give us some insight also in these fluctuations towards phase alignment, or like optimal phase and so on. But I'm trying in different ways, because obviously this moves from Lakatosh 2019, Nature Neuroscience, invasive recordings, beautiful study, but yeah, with EEG it's a bit different. But yeah, potentially attention.
Okay, thank you. That sounds great. I look forward to the new study.
Thank you for your questions. We stay online with your position and continue with Professor Dr. Joachim Groß, who is Chair for MEG-EEG Methods for Cognitive Neuroscience Research at the University of Münster in Germany.
Thanks very much. The candidate, congratulations to your thesis. I think you have completed a really remarkable and broad set of studies that extend all the way from research in healthy participants and healthy humans to patients and even for monkeys, and you even consider body-brain interactions. And I think you really address very fundamental and very important questions. And one of the questions, these fundamental questions, is about the tracking of rhythms in your first study. And it's an auditory study, and you're looking at delta and beta rhythms, and beta is often considered a motor rhythm. So you selected EEG channels, they probably pick up signals from auditory cortex, motor cortex. So why did you look at beta? What is your intuition of what beta is doing in this paradigm? And do you think it's coming from motor cortex? And if so, what is motor cortex doing in this auditory paradigm?
Deemed opponent, thank you for your questions and for compliments. Really, thank you for your question. I didn't have time to mention audio-motor interactions in timing processing, and so it's a good opportunity now. Didn't have too much time. So indeed, why beta? Because it's related to engagement of motor, pre-motor regions, and there is a lot in the literature showing that there is a fundamental involvement of such structures, and it's actually an extended motor circuits that involves, yes, motor, pre-motor regions, some more, if you want, prefrontal regions, but also subcortical network that also involves basal ganglia, cerebellum, and so on. In the lab, there is a lot of work on these subcortical structures, but also on cortical structures such as SMA. There is a theoretical paper on the role of low beta oscillations in such motor to auditory predictions. And there is then MEG as well as EEG evidence in simple auditory processing as well as speech processing, showing the fundamental role of motor to auditory type of predictions in timing processing and more generally auditory processing. There is even causal evidence with brain stimulation showing that if you act on motor cortices, for example, with TMS, with inhibition, you are then impairing altering perception or auditory processing. There is beautiful work from one of the opponents, Lars Rieke, and actually also Sanne with brain stimulation, also showing a role of specific oscillations in the tracking of rhythms, intelligibility of speech, and auditory discrimination. So I think overall, there is plenty of evidence on the role of these interactions, motor auditory cortex, with the idea that motor regions are engaged for top-down predictions about upcoming events. And so in a sense, they may be also resetting more bottom-up delta band activity from auditory cortices. In support of such view, we show that this beta band activity from frontocentral electrode, potentially from motor regions, it's superimposing a second level of timing that is actually these accentuations and fluctuations over time. So rather than processing a single tone over time, as you hear in the sequence, you are superimposing patterns of binary or triple or even mixed accentuations, which are not physically there in the auditory sequence. So in support of a role of top-down predictions from auditory cortices.
Great, thank you. Related to your comment on the accentuation, you show in this accentuation analysis that beta power is related to ERP, specifically to the ERP of the deviant stimulus. This is interesting because we had a study where we show in a study about sensory attenuation that beta power correlates with the M100 amplitude, and we relate it to prediction. So in your finding of the relationship of beta power and the deviant ERP, what is your interpretation? So what is beta doing to the ERP?
Thank you for the question. So I don't think there is this, or at least I didn't try to make a direct link between beta power and deviant processing. What I was trying to say is that, okay, we have this way of classifying trials where there are accentuations based on beta power. And not only we do see that these accentuations are present when we look at the standard tones, based on which there is the classification on trials, but this superimposition of accent actually influences deviant processing, on which we didn't perform the trial classifications. And so in a sense, it's not deviant detection per se, but it's this superimposition of accents, which is probably, saliency moments or, on-off-beat type of accents that it's influencing then the processing of a deviant tone.
Mm-hmm. Yeah, great, thank you. My final question, you show these different accentuation patterns and they change apparently throughout, across trials. So it's not only, let's say, the binary accentuation. Why would that be the case? So what is the strategy behind that? What would motivate a change of the accentuation pattern?
We even mentioned strategy. So indeed, is that a strategy? I mean, in a sense, it's not task relevant because the task doesn't instruct to impose accentuation for improving deviant detection. So indeed, how can we explain accentuations? I don't think that there is an answer in the literature. I don't think we provide an answer here, but I'm curious. I mean, obviously, because of chapter 5 and so on, maybe, bodily arousal states influence the superimposition of accents. I don't know. But the link that we try to make is between these mechanism of sampling or parsing inputs with some other type of functions. For example, grouping mechanisms that happens while, for example, speech processing. So this grouping of phonemes into syllables and so on. But it's a far type of link.
Thank you. Thank you.
Here, which will be continued by Professor Alex Sack. Here next to me, he's Chair for Applied Neuroscience here at Maastricht University.
Yes, thank you, Prorektor. Dear candidate, also my compliments for this wonderful and very inspirational thesis, also compliments to your supervisors. I was very inspired and I actually reached out to you after I saw it before I knew that I would be in your opposition because it got my attention. And I'm very interested in your proposal to consider the bodily rhythms and how they interact with the neural oscillation because that determines, in a holistic way, human perception, action, and cognition. But in your thesis, I see a lot of examples in the sensory domain. You play these groovy tone sequences, but I'm interested in cognition, and I really didn't get it yet. So maybe you can explain to me how, for example, in a working memory paradigm, where I try to understand the representation of items, multiple items, and we have a lot of ideas about neural oscillations and how the brain uses this temporal code to represent this, the theta gamma phase amplitude coupling and all these things, causal evidence even. Why and how would bodily rhythms add to explaining working memory performance?
Highly esteemed opponent, thank you for compliments and questions. Indeed, now we start another half an hour. So let me think of a short answer. Okay, chapter 5, the scope of chapter 5, it's opening to the new research line. So this is something that we have started last year, collecting some data. And so we have a whole paradigm with perception and action where we are testing the influence of body-brain dynamics on cognition in multiple aspects. And so it's all work in progress in Maastricht, in Münster potentially. Now, for working memory specifically, well, not necessarily working memory, but maybe memory, there is plenty of literature showing that there is an effect of the cardiac phase on the encoding and then memorization of items. In that sense, what experimentally you can manipulate in the study is when some stimuli are occurring relative to your cardiac phase. And there is an effect on systole versus diastole on potential memorization, learning, and so on, as well as there is an effect of attention and the likelihood of remembering items based on the cardiac activity. I guess there is also mounting evidence on the role of the breathing cycle, in the sense that some information are better processed when they fall on the rising versus trough of the breathing cycle. So I think there is actually a lot still to do. But if you want to have a a dynamic type of context to study, rather than very nicely manipulated experimental studies where when you're presenting the stimuli, then we should look at dynamics. And in that sense, you have this random presentation of stimuli, and then you model the likelihood of learning and remembering items based on what you decode being the phase of cardiac cycles or this arousal state based on breathing cardiac.
Can I just shortly interrupt you? I fully agree, and I would have no doubts that there are these dependencies between breathing and the cardiac phase or so and cognitive performance. I just wonder, how can they be independent from the brain? Because they must be top-down regulated by the brain, so the information is in the brain. If I have this information, I can just ignore these physiological signals, no? Or am I wrong?
Not really. I mean, there is also obviously, since there is mounting evidence on the role of body physiology, there is also more studies on the role of interoception, but also on the capability of processing internal states as determined by body physiology. And it's always about not only the switching between internal and external information. So there is a lot of work from Talon Baudry, for example, on this subjective frame and the capacity to switch between internal and external information. But there is also research on the capacity to actually bring this information, internal and external world together, and how that would influence would optimize sensory processing and perception. I think there is just a little bit more to explore, but not independent, but surely a source to explain variability in performance at the level of even the single subject.
All right, then maybe a last question where you can help me out with a study we did in Maastricht together with Tanné Engelen from Paris. Because completely independent from this, they were also interested to see whether these bodily rhythms, the heart, the brain, and also the stomach, so gastric, cardiac, and respiratory rhythms were measured continuously, physiological measurements, and then we investigated whether they would actually have an effect on the cortical excitability in the motor system as assessed by TMS MEP.
Right.
What do you think we found? The measurements are done. I just looked at the abstract of Tanné. So we looked at whether the phase of the cardiac, respiratory, and the gastric rhythm modulates the TMS MEP of the primary motor cortex.
Right.
What do you?
I would say it depends what analysis you used, so that I'm very broad.
Yeah.
But it also depends if you consider them together as defining a state, or if you consider the influence independently of each other, say gastric versus cardiac.
Independent from each other.
Independent from each other. So potentially there is one that influences more the cortex than others.
That's exactly right.
And it's probably either respiration or cardiac.
Yeah. No, what we found, it's very close. What we found indeed, which is surprising, that the phase of this rhythm modulated the MEP amplitude of the, there was clear modulator effect on the cortical or cortical spine excitability of the brain, but it was organ specific. So for a given subject, it could be either the respiratory or the cardiac, and it not necessarily didn't generalize to other organs. So an organ specific interoceptive profile.
Right.
Does it is that what you would expect, or?
Expect no, if I have to be honest, but the whole point of, one of the points of chapter 5 is indeed trying to find a metric to define arousal state, if you want, with bodily physiology considered together. So in that sense, okay, now probably there is one organ that it's influencing more cortex than others, but probably they are just, swapping, let's say, the order in this hierarchy of, driving arousal states, which is also something that we try to discuss in that paper. Probably it's not a fixed hierarchy, but it's, they are interchanging the role of defining arousal states.
Wonderful. Thank you very much.
Thank you.
Thanks for your questions. So we continue the position with Dr. Sanne de Nuva, who's assistant professor here at Maastricht University as well, with her expertise being in auditory and speech processing with EEG methods.
Yeah. Dear candidate, thank you for your presentation and your thesis. I really enjoyed reading it. Also, thanks for all your enthusiasm. That's very nice. Also, in organizing conferences, it's great that you're already doing this. I think I actually had some methodological questions I wanted to ask you, specifically on figure 2 of chapter 2. Because you find this very beautiful, strong intertrial phase coherence and a very strong ERP, which seems reminiscent of actually phase alignment and consistent phase alignment across trials. But then you go on to do, in some sense, the same analysis, but just changing the way you calculate phase, which is not using the Fourier analysis, but using Hilbert. And then you find not consistent phase alignment across trials. So could you please explain me this discrepancy? What am I missing here something, or why is this happening?
Yeah. Highly esteemed opponent, thank you for compliments and questions. Nice. So yeah, one of the problems, let's say, of intertrial phase coherence being estimated at the single trial level is that obviously the phase that when we are calculating coherence is the initial sort of phase at the beginning of the trial, based on also how we cut the trial, if we are padding and so on and so forth. Is that really a metric of how much phase aligns to tones and so on and so forth? I don't think so. So yeah, okay, ITPC is there, and it's also in chapter 3 and 4, because it's sort of the starting point. Let's reproduce what is in the typical studies in the literature, most established methods, FFTs, and so on and so forth. What 2B, and then in chapter 2, but also in 3, and potentially also the other work that we are currently doing, trying to find more time-resolved metrics to quantify if there really is alignment at the single tone level. And then we see variability. Okay, what is that variability? We have as mentioned before, there is a very long sort of time window. Here it's 60 milliseconds. Usually it's just 1 point, and there is an estimation of phase. So obviously there is variability because the time window per se, it's much longer. There is variability because there is more channels. Usually people focus on CZ, and CZ is the best signal. You get somewhat of a single phase, and that seems to be coherent over time. And here instead we didn't do that, and then we try to get coherence across a number of tones, 6 or 8. So I think what it's trying to do is just challenging the crowd, let's say, and stimulating questions. That's really what I'm trying to do.
Yeah, so I guess I definitely can appreciate there's variability, but to see nothing, it really puzzles me because the method is in some sense so similar. Yeah. But because you talked about it, because that's very detailed, I want to talk about this, seeing the consistency across a sequence, right? Yeah. Because in some sense, I wonder if you think this is really related to the rhythmic presentation, because if I would do the same analysis, for example, on ongoing alpha data and just pick every cycle, of course I also get consistent phase alignment. So to me, it just showed me that there's some periodicity at that frequency, but not necessarily that this is related to the rhythmic presentation, right? So Absolutely. Because you interpret it to me in a different way as if it's related to this rhythmic presentation, but that's really not necessarily the case because there is generally just autocorrelation in the signal.
I fully agree, and it could also be that there is actually an ongoing oscillations at potentially a slightly different frequency, and it's going independently of the auditory sequence per se. Mm-hmm. And so that variability that we see is just because we are sampling random points along the phase of these oscillations that are not caring at all about the auditory sequence. Mm-hmm. For me, it's totally a possibility. And so, yes, okay, we're making a claim about, timing processing and so on, on. But the real claim is that there is always variability with the methods that we use, and obviously then the description that we try to have.
Yeah.
So potentially, yes, they are independent. And that's also why we are trying to develop new methods to have a more sort of fine-grained idea, even like a method that allows to quantify alignment over time.
All right, yeah. I have a million more questions, but I give the word back to the project.
Thanks.
Yeah, thank you. So finally, the opposition will be concluded by Dr. Lars Rieke, Associate Professor of Multisensory Integration and Modulation, likewise here at Maastricht University.
Yeah, thanks, Rector. Hi, Antonio. Hi, candidate. Yeah, I think given the time, all the nice things have already been said. But I just want to second what Professor Groß was saying. I mean, this really popped out to me. I think you really showed a very high degree of versatility, so using all the type of methods that you have in this book, including even the theoretical chapter. So I think this is really, yeah, showing your broad expertise, and yeah, I was really impressed by that. So yeah, congrats to that for you, also for your supervisors. I have two short methodological questions, and I hope you can give me short, precise answers to this. So one is about the patient study in which you made, I think, in the abstract and also in the introduction discussion, claims about the causality. Yeah. And this is a word, that grabs my attention because it's very important, especially for not only for the mechanisms in the brain, but also for potential treatment. So can you briefly summarize what is causing what in that chapter, and how can you be sure that it's truly a causal effect and not just a correlation?
Highly esteemed opponent, thank you for complimenting the question. Causal because there is a focal lesion in a given part of the brain, and so if we observe any type of impairment in a performance or any type of behavior that we measure, then we tend to draw a causal link between lesion and impairment in that sense. Now, what do we show? Is there actually a causal role of basal ganglia cerebellum? We do see a causal role in the PS part of the PS, so in the production and synchronization, because we see that to a different extent, basal ganglia cerebellar patients do not synchronize to the external rhythms, or at least show much larger variability as compared to healthy controls. So in that, we are reproducing prior results, and we know that there is a role of basal ganglia cerebellum in different aspects of timing. So it's either temporal predictions for the basal ganglia, or more this interval type of processing that it's cerebellum-based. In EEG, then again, it's sort of the challenge. Can we actually test those capacity with isochronous sequences? I don't think so. You cannot disentangle this, a single interval. There is lots of work from Richard Ivory, Asaf Breska, and so on, or rhythmic sequences. And since I mentioned them, they have, much shorter sequences. They have 3 tones, and it's a rhythmic type of processing. In our case, can you actually call it rhythm? No, it's isochrony. Can you call it intervals, something else? I don't know. Mm-hmm. But surely what we quantify with this new method that we are trying to introduce is that they are very different from the LT controls in a way or another.
Okay. And how can you be sure that it's truly an effect directly on the temporal processing? Because, the lesion could also result in changes in any other variable that you didn't measure, which then leads to the change in processing that you observed.
Absolutely. That's an open question, I think. In that sense, we don't measure anything else, at least in this study, except from, these timing sort of processes. Mm-hmm. So, we just draw a conclusion on how it affects these capacity that we are testing, but I wouldn't know if they affect anything else.
Okay, good. Thanks. And I think I still have 2 minutes. Okay, so then also very briefly connecting to what Dr. Keitel was asking about, I think it was chapter 2 where you showed the phase analysis and the tracking analysis, and you attribute this to predictions. And I think prediction was not even a variable in your design. There was an isochronous sequence, but it was not a parameter. So I'm wondering, how can you draw a conclusion, and how can you link this to a constant, putative constant in your design?
Completely agree. I think the predictions are in the accentuations. In the sense, these accents are not there in the physical stimuli that you're listening to, and anything else that pops out in brain data and it's not in the auditory sequence per se, it's an effect of prediction. So any type of top-down influence on how you are potentially perceiving these sounds, although we didn't test perception. When it comes instead to the rhythm tracking, indeed, is it predictability? Is it memory-based type of processing? But yeah, not timing predictions per se.
Okay, and I saw, so you said you are working on this in a new study now where you actually monitor the attentional state and of the participant.
Not only the temporal state, but also in the new data collection that we are doing, we have a manipulation of the auditory sequence. So there are moments of predictable speeding up and slowing down where you know where you can start actually establishing prediction, and it influences the tuning in and out from the auditory sequence.
Yeah, very good. That's what I needed.
In time.
Antonio Criscuolo, the time appointed for defending your thesis has now passed, and the degree committee will now withdraw to discuss the quality of your thesis in your defense, and request that you and your company wait here in this room until we return.
Thank you.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Antonio Criscuolo, the degree committee here present has assessed the quality of your work and your defense, and in view of the positive verdict and taking into account your previous qualifications, the committee decided to grant you the degree of doctor. Professor Kotz is now authorized to confer upon you this academic distinction in accordance with Dutch university custom, and invite your supervisor to now take the floor.
All right, please. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and reasonable?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Antonio Criscuolo, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and other members of the committee, and affixed with the official seal of the university. Dear Anto, dear Dr. Criscuolo, veni, vidi, vici. Following good ancient Latin tradition, you did conquer all hurdles of this PhD, and in a very special, wavy way. Also on behalf of Michael, I congratulate you and your whole family to the great scientific achievement. I would like to take this special moment and look back to an event that you in your own way called, on a random day in October, I had the urge to speak with a stranger about her research, and she let me walk into her office. In you walked, and we never stopped discussions about the depth of the human capacity to use and understand time. It seems that since when you walked into my office, often with Michael, many times, what we talked about was not my research, but what became your research or our research on time and rhythm. You took on this complex topic for your PhD research and made it your own, riding the waves of brain oscillations and sophisticated algorithms. But let's reflect for a while on what makes an impressive Italian scientist with a spicy twist. And what better way than thinking about a recipe, especially for someone who loves to cook. So get ready to sprinkle some incredible energy, whip up some waves, and toss in a whole lot of creativity. This recipe guarantees to cook up a scientist with a flavor that is as vibrant as the Salerno sunset and as tasty as Anto's Nonna's Mariana sauce. Let's start with the ingredients. First, we take 1 large cup of energy. Start with a generous helping of enthusiasm. This ingredient will fuel any scientific endeavor and keep you buzzing with excitement, just like a shot of espresso after a big Italian meal. Then take a few tablespoons of waves, add a dash of determination, and mix it with ambition until it forms a peak wave. This allows you to tackle any challenge like conquering a mountain of data or your supervisor's persistence on certain formats. Then we add at least 1 pinch of Southern Italian creativity to that as extra flair. Think outside the pizza box and let your imagination run a little wild. Not to then miss adding a healthy dose of independence at will. This ingredient will give you the freedom to explore new ideas and chart your own path, just like an olive tree standing tall on the terraced hills that reach down all the way to the Tyrrhenian Sea. Oh, and then we need a good tablespoon of organization. Mix in skills to connect with fellow scientists and spread your scientific gospel far and wide, like the WAVES conference and the WAVES summer school in Salerno. Just like setting a traditional southern Italian feast, bring everyone to the table and let the ideas flow on the beautiful Amalfi Coast. Last, sprinkle in some personal baking and pasta making for the extra Italian touch. Whether you're whipping up a batch of fresh pasta or baking a delicious tiramisu, let your culinary skills shine through in your scientific endeavors. Until what would we do without your passion for food that you share so generally with all of us? Now, if you want to cook this recipe, follow the following instructions. Preheat a scientific oven to 350 degrees of curiosity. In a mixing bowl, combine energy, waves, creativity, independence, and organizational skills. Stir vigorously until well blended and bubbling with excitement. Folding in the baking and pasta making with care, making sure to incorporate every flavorful morsel. Transfer the mixture to the laboratory and bake for 30 minutes or until golden brown and bursting with scientific brilliance. Serve hot with a side of passion and watch as your scientific career rises to new heights. So there you have it, the recipe for a successful scientist with a Southern Italian twist. Buon appetito and happy continued experimenting. Dr. Criscuolo will be at your side.
Dear Dr. Criscuolo, also on behalf of Maastricht University, I would like to congratulate you on the degree that you have acquired, and I include your parents and company in these congratulations. I now close the session. I would like to ask, before we proceed to the reception and the refter, to give the online opposition members the opportunity to congratulate you before we take a quick picture here. Dr. Keitel, if you wish.
And I look forward to discussions with you.
Could you repeat that? You were muted, unfortunately, here on our side.
I'm unmuted.
Now we hear you.
Okay, now, okay. Okay, what did I say? So probably congratulations, Antonio. That was really, really well done. And congrats also, your family can be very, very proud of you. You can be proud of everything you've achieved. And I look forward to discussions with you.
Thanks.
Dr. Gold.
Yes, Antonio, also congratulations from my side. I think you've done an excellent thesis, an excellent defense. You can be really proud of what you have achieved with your thesis. You have a broad and fascinating research profile, and I'm very much looking forward to your next studies and to your new ideas. So congratulations, and today is a day to celebrate.
Thank you.
Thank you. Thanks again for joining remotely. So now I invite everybody to leave the aula and proceed to the refter for a little drink and snack and maybe continue discussions. And we shall stay behind here for a quick picture and then follow you. Thank you everyone for joining.
Recording stopped.
Yeah, we take pictures now. No,